Well, good morning. We're going to go into the afternoon. My name is Nikki Baker. I work with the New Jersey Work Environment Council as the Healthy Schools Now organizer. So we organize for health and safety around schools, especially safe school buildings and facilities. I have the pleasure of moderating this wonderful panel of researchers. I will briefly give an introduction, but I'll let you introduce yourselves in some light. So we have Matt Campen, a professor from the University of New Mexico, and he has a focus in systemic impacts and neurovascular environmental impacts. We have Phoebe Stapleton, also a doctor who works in maternal and environmental impacts with maternal and fetal impacts. And I am shortening these resumes because they are quite long. And Phil Democracy, whose research on nanoparticles and biological environmental systems far goes impact. So I'm going to let you do yourself more justice than I can, and thank you for coming. Matt's going to come up first.
Thanks.
Now we do have slides.
I have slides, and I'll put through them. Thank you for the opportunity to talk with the community here. I got a chance to give more in-depth science yesterday for the Rutgers group, and this has just been a great visit for me. I'm out of New Mexico. I want to make a contrast to that. We're here in New Jersey. This is the #1 population dense state in the country. 1,200 people per square mile. New Mexico is 45th, 17 people per square mile. Obviously, most of them live in my town of Albuquerque. It's not that absurd of a rural or frontier area, but it does speak to, I want to keep that in mind as I go through the data that I show, because they're from humans, largely, that are from New Mexico. And the human impact on that environment is by magnitude is going to be less than what you have here in New Jersey. So in some sense, the data I'm going to show you are sentinel, and things may be different here. So, that's my kid from 5 years ago. Part of my story, getting into plastics research, began with some 7th grade middle school science fair projects that ultimately got shut down. We collected the school data and we got shut down because of the pandemic, and we didn't get to present it, but I've used that to build sort of a small research empire, and I like to continue his story as I talk about this. This is the Rio Grande Reservoir. This is very close to the headwaters in the San Juan Mountains of Colorado, where the Rio Grande forms. The Rio Grande is by many definitions a very small river. It doesn't have a lot of flow. It actually dries up south of El Paso and then reforms down in Texas in the Big Bend National Park. Its cultural and agricultural relevance is massive to our region. It is the river. So in that sense, I point out this building I passed many times traveling onto the Navajo Nation. Water is life. When you live in the desert, when your community and your sort of cultural background stems from preservation and utilization of a really finite resource, you are very drawn to the relevance of what little water, what little drainage we have. So here's that reservoir, and this is a map. It might not have a lot of perspective for everyone here, but this is northern New Mexico from Albuquerque up to the border with Colorado. And this is the river, and it goes up into Colorado. Just west of Creed is where it forms in the mountains. There's a lot of mining going on, or historically in this area. It flows from this point where we collected our first samples. Upstream of there, only about 700 people even live, so very little human impact. And then at the second point, Taos, New Mexico, we sampled again. And this larger dark blue area is the watershed And about 70,000 people live in that area, San Luis Valley through Colorado. There's a lot of agriculture up there, a lot of livestock, but relatively limited human impact. Only about 70 to 80,000 people contributing to the watershed at that point. And then you get down here to just south of Albuquerque along the river, north of the Isleta Indian Reservation here. And now, all told, there's only about a million, there's now about 1 million people. From a New Jersey standpoint, I know that's pretty limited. That's more than half of the people in New Mexico. So you get a feel for this. And just to point out how important this river is, each of these red dots, these little red crosses, those are Native American pueblos that are clearly not randomly assorted throughout New Mexico. They are along the Rio Grande. Its historic relevance is very clear. So my son collected this water. He collected it in glass bottles to make sure that there were no plastics contamination from our process. We used a filter in our lab. Obviously, he got help from Dad. But he then was able to use a black light that helps fluoresce the plastic particles, and we were able to identify and count the particles. So pretty straightforward. And what he showed was that from the reservoir up here, going down to Albuquerque, you can see increasing amounts of microplastics as you have greater human contribution to the watershed. That didn't concern me nearly as much as the fact that it was really easy for a 7th grade researcher to see particles in one of the most remote parts of our continental United States. So this dragged us into this world of asking, are plastics really bad? I never would have thought they're bad. I've been studying inhaled pollutants for a long time. I've studied sarin gas. I've studied botulinum and diesel exhaust and coal and all these things. So plastics, we implant them in our bodies. They're super inert. Your body doesn't respond. You can have plastic parts in your hip. You can have them in your blood vessels. So it took a lot for me to start caring, to be perfectly honest. But knowing how easy it was to find these, we start looking at the reality, which is this is exponential growth. Our planet, not just America, not just New Mexico, not just New Jersey, our planet has been generating and using plastics at an exponential rate. Basically, the amount on this planet doubles every 10 to 15 years. That is slowly degrading. Dr. Lumbach showed wonderful images, horrific images of the landfills. Most of what we're doing, we're just throwing away. It doesn't stop existing. It breaks down slowly into microplastics and then nanoplastics, and the curve of the generation of microplastics completely mirrors this exponential growth. So we have this problem. We think we've thrown it away, we think we put it in a landfill, but we haven't gotten rid of anything. So this is the process. You have a macroplastic, a jug, a car dashboard, your phone. We're all part of the problem. It's not something. It's not there's no otherism here. We're all part of this. It breaks down into smaller pieces through oxidation and sunlight, heat and weathering, and it gets smaller and decrepit. The microplastics are what we focus on because we can see these little tiny dots. We can look under a microscope and see these little tiny dots. And after that, they become nanoplastic. And I'll show you some images with an electron microscope that might help redefine what it is we're worried about. Because maybe these microplastics are actually kind of huge. This scale, I love this. This is from a laboratory in Australia, and I asked him if I can use this image, and he was just thrilled that anybody would have asked, because he could just rip it off the internet, right? But he describes, so this is a tennis ball. It's about 10 centimeters in diameter. You've got salt grains that are about 1 millimeter in diameter, and then you get down 10 to the 3 nanometers is 1 micrometer. So when we say microplastics, we mean things larger than 1 micrometer, but smaller than 1 millimeter. And the size of our cells can be anywhere from 10 micrometers up to 60 to 100 micrometers. That's sort of what a cell is. And so a lot of these microplastics are the same size as your neurons, your immune cells, your cardiac cells. When you get below 1 micrometer, now you're talking about 1 to 1,000 nanometers. And now you're talking about the size of viruses. COVID was about 50 nanometers. I'm going to show you images of plastics that are about 50 nanometers, 150 nanometers, 200, 300 nanometers. But these are now things that can get inside of cells. And we're having a lot of trouble finding them because they're so small and so difficult to observe within the human body. If I leave you with one thing, the media pushes this sort of narrative that, oh my gosh, I'm getting microplastics from this bottled water. Yeah, I mean, I don't want to encourage use of plastics when you don't need to. By no means do you need a plastic water bottle. Use glass or reusable thing. But this is not really the thing. What happens is this bottle is going to be thrown away, and in 40 years it'll become nanoplastics. And think about it this way: if you took that water bottle and you emptied it and weighed it, and then you filled it 1,000 times over the next year, and you came back and you weighed it again, we can agree it's going to weigh about the same, maybe 99.9998% of what it used to weigh. So that 0.00002% is maybe all that became microplastics, but all of it will eventually become microplastics. So that's sort of this weird narrative that we have to deal with from the media. The media is a double-edged sword on this. And what I worry about is that we're focused on things, we've forgotten about this stuff. We've thrown it away, we put it out of our mind. We can't do that. So my group has been doing these studies. I'm a standard toxicologist. I take toxins, particles, air pollution, and I put it onto cells and culture, and I look at their response. I use mice, we expose them. The problem with plastics is we don't know how much to expose the animals to, what is relevant. And so this started sort of this long, painful journey. I have a wonderful team of scientific experts who kind of all hate me now for taking them on this journey, because it's been so difficult to just learn new things and develop new techniques. But we need to figure out a way to pull plastics out of humans and look at them with analytical chemistry techniques. The way we've done this is if you've seen the show Breaking Bad, you might know what it is we do in New Mexico to get rid of a body. We use chemicals, and so we've been working with Office of the Medical Investigator to obtain decedent samples. So these are from autopsies. They're parts of the human body, including the brain, liver, kidney, testes, and we can digest those in very caustic chemicals. Potassium hydroxide, it's a very powerful base, and it digests most of everything away. 99% of the massive tissue goes away. We can then centrifuge and pull out the solid particles and then run those on a mass spectrometer and get a very nice signal and a quantification of how much plastic was from that sample. So it's stepwise, and it's been something we've developed over the last 2 years. It's not perfect yet, but we're continuing to improve it. This was our first foray into this. Marcus Garcia is a pharmacist, researcher who joined our team as a postdoctoral fellow. He's really led the way in developing this technique. And our first work was with Baylor College of Medicine from Houston, and they provided human placentas. So this is a discard tissue. It's easily donated for research. They have a bank of 5,000 of these in a repository. And we started with this. And what we found was, so each of these little columns is an individual donor, and the different colors represent different polymers: polyethylene, polyvinyl chloride, polypropylene. And you can see that there's a wide distribution from person to person. And having stared at this data now, I'm a little bit numb to it. I would actually say, thank goodness most of them are quite low. When we first looked at, the raw opinion when we first saw this data is, wow, we can find this in every tissue we look at. There's no hiding from these plastics anymore. For the most part, these are barely at the levels of detection. And we're starting to be concerned with what's going on up here. We do have some hints that it might be the things we eat in our diet. There might be some relationship with outcomes of gestation, you know, like premature birth or gestational hypertension, and we're looking at that. But we literally, this is 60 samples. We've now analyzed 200 samples. To get to 5,000 is going to take us a while. We also simultaneously were running these studies with John Yu, pictured here. He established a relationship with veterinary clinics throughout the Albuquerque region. So he's interested in how plasticizers, we talk about phthalates and bisphenol and things like that, how do those chemicals affect sperm quality and fertility? And so he works with the veterinary clinics so he can use dogs as sentinel animals, because humans, we don't willingly give up these parts, right? So these are testes from dogs and from the autopsy specimens I talked about. The first kind of crazy part, I mean, this is on a log scale, but this is basically dogs have 1/3 the amount of plastics in their testes that humans have. Even though dogs eat off the floor and chew on little plastic things, it really speaks to what is probably in our diet and the process by which we grow food and sell food and eat food. That was pretty alarming. One of the kind of good things I will say from this study is this is a graph down here showing the relationship between the amount of plastics in the testes of humans and their age. And it doesn't just keep getting worse, right? The body can clear these, is our conclusion. The body will absorb these and clear them. It's a slow process. It's not like, you know, taking vitamin C. If you know, it's sort of a joke in our field in the world of pharmacy and supplements that if you eat a lot of vitamin C supplements, you're basically creating the most expensive urine you can, because it's just coming right through your body. These are more like months to years to build up and then steady state level off. If you could stop eating the plastics, it would probably start going down. The problem we have is that exponential growth in the environment. And so even though we're happy with the steady state of uptake, the dose keeps going up for us, and so our equilibrium is going to keep doing this. But anyway, this is just some of the conclusions we can draw from our early work. This is a study that is now in a preprint form, which means it hasn't completed peer review process. We're going through it, but I can say that the outcomes I'm talking about now are stable. They're not changing. This has drawn a lot of press as well. But we obtained liver, kidney, and brain from autopsy samples, about 20 to 30 per organ. We obtained liver samples from 2016 and 2024. Our original agreement with the Office of the Medical Investigator was you're going to be throwing away these tissues anyway. After 7 years, it's a forensic requirement in case there's some legal action that needs to take place for a given death. So we were able to get those, and then after we saw what the data looked like, we said, all right, it's now 2024, give us sort of an equal number of recent samples. We went through compliance, we got consent, and we were able to get those. Ultimately, the upshot of this is the liver and the brain are both significantly increasing. I keep losing my little asterisks that indicate significance. They've gotten about 50% worse over this 8-year period. The kidney hasn't changed, which is interesting. But the other issue is that the brain has 10 to 20 times more plastic in it than any organ we've looked at. There's something unique about the human brain. As I mentioned, we've been throwing away plastics. We've been trying to put them out of our minds. They seem to be working their way right back in. Other things I can say about this data. People that come to the Office of the Medical Investigator represent a very wide swath of our region, in the sense of, you know, there's natural death that's unexplained, there's a lot of traumatic death, there's a lot of drug-related deaths in Albuquerque. So this is as random a sample as you can imagine. And I'll just leave you with some of the images that we've been taking, because it's tough to take this mass spec data and really create the awareness and the understanding. But these are histological sections from different organs that we've sampled. This is the kidney, and these little bright spots, we use a reflective polarization wave method of sort of seeing the refractile nature of the particles that we see, and that's a strong indicator. It's not 100% confidence that it's plastic, but you can see they tend to show up in specific regions of the organs. This is called a glomerulus. It's where the blood of your body goes through the kidney for filtration. And hey, that kind of makes sense. Here's a bigger one in zoom. It's both a beautiful, beautiful, horrific histological image, but it clearly shows that where the blood is coming through the kidneys, it's getting cleared. And that seems to be where we see the most of these. That's a good thing. Like, our bodies are not just accumulating these if we don't have to. This is the liver. What's interesting in the liver is the liver has little fatty lipid droplets, and we tend to see these almost beautiful kaleidoscope images here where these longer crystal-like structures are accumulating. And this is the brain. The brain's interesting because we don't see those collective plastics. They tend to be more spread out, and they tend to be smaller. But we can definitely see these throughout the brain. We don't really see a pattern like we see in the kidneys. So our group is diving in and trying to understand this better. These are electron micrographs showing the liver, and here's a particle in the brain. But this is the real eye-opening part of this for us. Okay, you can see these those were micron-sized particles that we can see with a microscope. Light microscopy fails below 1 micron. When we get into nanometer size range, the wavelength of light is about 400 nanometers. So the waves of light just sort of pass through these things. We have to use electron microscopy to see things this small. So these are particles that we isolated from the brain, and we spread them out, and you can see these things are little flakes, little shards of plastic that are about 150 nanometers long by 40 nanometers wide, and they are not much bigger than viruses. And they are replete throughout all of the organs of our body. So how do we know they're plastic? We use spectroscopic imaging to confirm this, but we did this one little experiment. We took these particles and we heated them to the melting point of polyethylene, and you can see how they start to fuse together. They melt at the right temperature for polyethylene. So I'll close. We're late to the game. We've been doing this for 50 plus years. Our agricultural system has been using plastic, has been using water that's contaminated with plastic, and we have this feed-forward biomagnification problem, so that all the food we're generating in this country is building up, and we've measured it in ham and beef jerky and chicken. We can see this stuff. So we're late to the game. We have, do I know that this is bad for our health right now? Deep breaths. If you can avoid a lot of high-risk behavior like base jumping or bicycling the wrong way in traffic, in this country you should live to be 80 years old, 90 years old. Plastics are a part of that. Plastics, I can't imagine administering an IV drug without plastics. I can't imagine a colostomy bag or sterilization techniques in the hospital. I have a very close clinical affiliation with my job, and plastics are a part of healthcare, food transportation, food preservation. They are something we need, but we need to do a better job of taking care of it, making the transportation safer. We need to make the waste management more effective and figure out a way to really turn these into something that's safe. And I'll stop there, and I guess Phoebe's next. Where's Phil next?